Thank you, Sue, and good morning, everyone. Thanks for joining our webinar. So I'll start with a short intro about Volition and then hand it over to Andy and add a few more words about our commercial strategy at the end. So first, Volition. Our mission is to save lives and improve outcomes for millions of people and animals worldwide. We are a diagnostic company focused on epigenetic marks. We have different types of application on the human as well as on the animal side, screening, monitoring, disease monitoring, and you'll see that in our presentation and the clinical use case that Andy will outline. We started our commercial journey and our monetization of our IP. We believe our addressable market is very sizable. We started with vet, and we are entering now into the human space. We have two key areas where we will be focusing in the short term. Vet, so we have our cancer product. Now we are working towards scaling this product. And then NETosis. And on the human side, obviously, we have the NETosis product line that we're going to discuss. We also have something that is a little more on the early side, but I think it's important to mention it quickly. It's our licensing opportunity on the oncology around the CTCF chip technology. So now I'll pass on to Dr. Andy Retter for his presentation, and I'll come back quickly at the end.
Hello everybody, and thank you to Gael for that introduction. My name's Dr. Andrew Retter. I'm an intensive care consultant and specialist in ECMO and thrombosis, and I'm here today to take us through sepsis and how it relates to our products and H3.1 going forward. I am going to cover some of the stuff I've talked about previously, but we do have new information to share with you today. Just want to highlight my potential conflicts of interest. I'm really passionate about sepsis. It's my clinical focus, my research focus, and most importantly, I'm desperate to try and improve. Along with Volition, we share a passion for trying to improve outcomes of patients with sepsis. The views expressed here today are my views, and they do not represent the NHS Foundation Trust I work for in any way. Sepsis derives from the Greek word sepo, literally meaning I rot. If you go back in history, we can see that the Egyptians recognized patients with sepsis, as well as the ancient Greeks. Roman literature comments on it as well. But it wasn't really until just before the First World War that traditional definitions were starting to be described, and then more recently work in the 1980s, 1990s, early noughties, right up until 2016, work has continued to refine the definition of sepsis. The 2016 definition of sepsis is a real step change. It talks about a dysregulated host immune response for the first time. And really, this is where we're targeting the H3.1 test to try and identify those patients who are sickest and are most likely to deteriorate. Sepsis is a life-threatening condition which arises when the body's response to an infection injures its own tissues and organs. Our immune systems evolved to localize and try and wall off infections. And so the immune response is incredibly precisely controlled. It's exquisitely localized to the site of infection and to the time of infection as well. So the changes that happen are just really spatially controlled. What we're starting to think now is that sepsis, at least in part, relates to this loss of localized control. And the excessive immune response that's generated ultimately damages other tissues and organs. Sepsis is indiscriminate of age, ethnicity, and socioeconomic status. It can be triggered in otherwise healthy people, but people with weakened immune systems are particularly at risk. And given the increasing number of chemotherapy agents and novel cancer drugs, the number of patients vulnerable to sepsis is ever increasing. Sepsis can be thought of a disease of the very sick, but 80% of patients develop their infection outside of hospital and present to the emergency department or emergency room as the first step on their journey into hospital. Sepsis is a spectrum which evolves over time. Sometimes the deterioration is rapid and sometimes the deterioration is slow. And this is again where we think H3.1 will be very helpful in identifying the trajectory patients are following. Sepsis is one of the most preventable causes of death worldwide. There are almost 50 million cases a year, resulting in around about 11 million deaths. It's responsible for just over 40% of the deaths in children under the age of 5. In the UK alone, 5 people every hour die of sepsis. That's more deaths a year than bowel cancer, breast cancer and prostate cancer combined. What's even worse is that 40% of the survivors have long-term consequences. The most obvious changes are people can lose digits, fingers and toes, but many describe cognitive impairment with impaired memory and concentration, decreased function, decreased exercise tolerance, inability to return to work, and sepsis is a huge cause of lost work hours in the workplace. Despite this overwhelming burden of disease, sepsis receives relatively limited funding compared to other comparator conditions. We would argue that it's a great unmet population health requirement. You can see from this slide provided by Professor Djali Anand that the incidence of sepsis per 100,000 of the population is 377. Again, that's more than heart attacks, strokes and cancer. But you can see the funding. Cancer attracts by far the most funding, and yet this disease, which is preventable with the correct treatment and identification, in the majority of cases, receives by far and away the smallest amount of funding, at approximately $91 million a year. Sepsis is the great mimic. It's extremely hard to diagnose, and indeed the 2016 definition of sepsis that I mentioned a little while ago talks about the concept of occult sepsis or hidden sepsis. People can be fine until very late. People don't always have to present with a high temperature. Indeed, a low temperature is probably suggestive of more severe disease. Sometimes people just present confused. Sometimes people have a cough and a raging fever. Sometimes patients just have difficulty passing urine. Sometimes people fall over. Given the diversity of symptoms, their vague nature and the inconsistent way they can progress, our hope and the design behind our studies is to try and produce data which will enable clinicians to identify which patients are more likely to deteriorate and those who are more severely ill. We hope that this will enable us to start treatment in a more timely fashion, which will lead to improved outcomes. The risk of death from sepsis or from bacterial sepsis increases by 7.6% for every hour in which appropriate antibiotic therapy is delayed. That's an amazing figure. What we should also explore is there are viral and fungal causes of sepsis too. And so there's a huge amount of work to do to try and improve outcomes. Our current diagnostic strategies are empirical and often involve choosing best guess antibiotics. Research tools have been developed to help us identify and track the care and outcomes of patients with sepsis. These are classically the SOFA score, the SAPS-2 score, and Apache scoring systems. However, I'm really passionate about sepsis. I see a huge number of patients with sepsis. We don't really use them in our day-to-day practice. They're slow, there can be errors in adding them up, and they're a bit cumbersome to use. They are very useful for research studies in identifying the sickest patients and making sure that we've recruited the right people, but they are not really used at the bedside. In medicine, as in life in general, we're often trying to oversimplify things. And rather than sepsis presenting as one point in time, it's a spectrum that evolves over time. I've stolen this graph and I've modified the subsequent slides from Professor Hodgkiss's paper, which was published in Nature Review of Disease Primers in 2016. And basically it's trying to show the evolution of the immune response over time. The prevailing thinking is that when we have an infection and our cells are damaged, a huge number of proteins leak into the blood, which can stimulate an immune response and ultimately lead to the body attacking itself. And therefore, at this point, the immune system is downregulated. And so what you can see is we oscillate between a hyperinflammatory phase and an immunosuppressive phase. That's why in this graph there is a middle line where patients' inflammatory response waxes and wanes over time. Our early data suggests that H3.1 may be able to help us identify this waxing and waning nature. Going forward, we hope that H3.1 will enable us to determine therapeutic strategies to try and interdict as patients become hyper-inflammatory, to try and understand those who are sicker and hopefully develop new strategies to improve patient outcome. Pulling these last few slides together, we believe that sepsis represents a huge unmet medical need across the whole population demographic. There's a desperate need to develop new diagnostic tests and new therapeutic strategies. Diagnostically, we can differentiate two groups of tests: those that help us identify the presence of bugs, microbes, bacteria, fungi, viruses, and those that help us identify the host immune response and study the host immune response and potentially lead to modification of the host immune response. If we pause for a second and look at COVID, many of the strategies in COVID manipulated the host immune response. Steroids from the REMAP-CAP study, tocilizumab also from REMAP-CAP, baricitinib, all attenuated the immune response. And if we can identify strategies to provide a more targeted and precision approach to the management of sepsis, we really hope that we'll be able to improve outcomes. At present, the test takes 45 minutes. We're well on the way with work to take this down to 20 minutes. And therefore, we potentially have a test which can be run repeatedly to follow patients' trajectories and aid in their diagnosis. We believe this will be a game changer to help clinicians at the bedside monitor the progress of sepsis patients. I'd like to try and bring sepsis to life by walking you through a recent clinical case. This case involves a 34-year-old gentleman who actually caught flu from his daughter. He was otherwise fit and well, healthy, walked miles a day as a postman, smoked 5 to 10 cigarettes a day, and he felt unwell. Felt unwell with a high temperature, flu-like for 2 or 3 days, and then presented to his GP, had a very high temperature, and was sent to the emergency room. He rapidly deteriorated. By the time of his arrival in the emergency room, he was blue, proper name for that is cyanotic. His oxygen saturations in his blood were very low, and his chest x-ray showed infiltration in both lung fields, and he was extremely unwell. Shortly thereafterwards, he was intubated. That's to say, a breathing tube was placed in his windpipe in the emergency department, and he had lots of lines put in to give him medication to support his blood pressure, to help his heart, to help his kidneys, and obviously to give him antibiotics too. You can see here on the x-ray the shadowing in both lung fields, just consistent with the infection and with fluid associated with that. And this is flu. And this is classic flu presentation in a young patient. He also had a secondary infection with something called Group A streptococcus and was very, very unwell and profoundly shocked. He was admitted to intensive care. He continued to deteriorate and we were no longer able to safely ventilate him. That's mean that the level of oxygen we were using and the pressures we were using to get oxygen into his blood were so high that they were actually damaging his lungs. And so we placed him on ECMO, extracorporeal membrane oxygenation. It's essentially a mini lung bypass machine. We spent about 10 days on ECMO and a total of 14 to 15 days in the intensive care unit. We were able to take the breathing tube out successfully, and he was stepped down to the ward. We spent another 3 weeks of rehabilitating, getting stronger prior to getting home. He's still not back to work. This is a young, fit, healthy man previously, showing the effect that sepsis and infection can inflict on people. He actually lost a few toes. You can imagine a postman losing his toes and having difficulty and pain walking has profound life-changing consequences for him and his family. If we can help identify these patients sooner, hopefully we'll start treating them sooner. H3.1 may give us a new therapeutic target too to improve outcomes, and so extremely hopeful for its use and adaptation in the future. In the next few slides, what I'm going to do is explain how H3.1 works, and I'm going to try and tie it together with our immune response and how when that immune response goes a bit awry, you can get organ damage. And I'm going to tie it up with the clinical cases we just mentioned. This slide here shows a healthy neutrophil in the bloodstream, just circulating. Upon exposure to a bug, it becomes activated and it releases these neutrophil extracellular traps behind it. These nets are decorated with proteins that are toxic to bacteria and viruses and fungi. The proteins, some of these proteins also signal to other immune cells to say, come here and help guys. And then these nets actually act as sort of a physical scaffold to trap some bacteria. And we know actually that a number of bugs can manipulate nets as a way of trying to survive a bit longer. If we think about our postman had a very severe pneumonia and severe chest infection and activation of the immune system. And his immune response was such, and his organ failure was such, that he actually developed kidney failure and heart failure. And if we think about it for a second, it's a bit odd. Someone's got a low level of oxygen in the blood, but we correct that with medical therapy, with a ventilator and extra oxygen, and we're treating the infection. But why do their kidneys fail? Why does their liver fail? Why does their heart fail? Why do they get confused? And it's very complicated, that immune response, and H3.1 is a small part of that. But if we can identify that and understand that more, that's really useful as a diagnostic and therapeutic area going forward. What happens when nets become excessively activated is our endothelium can be damaged, and we can get loss of the localization and coordination of the immune response. And that's why you can see organ damage to these tissues below, to your heart, to your lung, to your liver, kidneys, to your bowel and to your brain. A true multiple system problem. Volition's test, the H3.1 assay, is picking up nucleosomes, which are a key building block, a part of neutrophil extracellular traps. Nucleosomes are DNA wrapped around histones, and you can see here the antibody test specifically picks up histone H3.1 and then has a second binding motif which makes it specific for nucleosomes. Our research has shown that this correlates extremely well with neutrophil extracellular traps. So, for the first time, we have a test which can potentially show activation of our innate immune system, which can be scaled rapidly, which is cheap, and can be delivered in a meaningful time frame. It's 45 minutes to run this test, and we hope to get it down to 20 minutes very soon to show clinicians what's happening and give us a window into our immune response. Outside of PhDs and thousands of pounds on complex research tests or thousands of dollars on complex research tests, we've never been able to do this before. And that's why we hope that H3.1 is potentially game-changing. Just to preempt some questions, how do we know that neutrophil extracellular traps are essential in sepsis? Well, we've got a large amount of experimental data from animal and now from some human models suggesting that elevated levels of cell-free DNA and elevated levels of netosis are found in patients with sepsis. The highest levels are found in those patients with the most severe sepsis. And there's increasing data to suggest that the high levels are associated with a worse outcome. How can we show that H3.1 is an excellent marker of nets? That's been a really key research question for Volition. And we have just submitted a paper working on a novel synthetic model of sepsis. It's a model of sepsis in a test tube, basically. And I'm really excited to say what we've shown in that is that we can use biological triggers to stimulate neutrophils and model sepsis. And we've shown that H3.1 correlates extremely well with neutrophil activation. And so we have now got clear scientific data showing that H3.1 directly links to neutrophil activation. Do we know that removal of nets improves outcomes in sepsis? There are two potentially extremely exciting areas where it's been shown that removal of H3.1 nucleosomes potentially leads to improved outcomes. It hasn't been done in humans yet, but there is a pig model of sepsis using a immunoabsorbent column. What does that mean? It's a column of blood outside the body that blood passes through, and it's lined with antibodies to capture and pull out nucleosomes. And in a pig model of sepsis, that improved the outcome of pigs. Very recently, a paper has been published on a sheep model of sepsis. And in that model, a specific drug was given to bind to and attenuate the effect of circulating nucleosomes. And again, that paper showed improved outcomes for the animals with sepsis. They required less organ support and they had less of an inflammatory response. To me, that's really, really exciting because you've got two independent methods by which we've lowered H3.1 levels and shown improved outcomes. It's not absolute proof, but it's a really great insight into a new potential therapeutic target and shows the huge potential benefit of this test in identifying patients, in monitoring patients, and actually targeting their care. We have several large-scale clinical studies underway where we're working with a number of key opinion leaders and centres of excellence across the field. And we hope to have more concrete data to show at conferences and publish later in 2024. Our aim is to launch a low-cost, rapid, easy-to-use, accessible diagnostic test, which is affordable not just to first world, tertiary referral, high-income specialist hospitals, but is accessible everywhere. Sepsis is a global disease, 50+ million people a year, and so our aim has always been at a very broad market, not just teaching hospitals. We hope that H3.1 will enable us to identify and personalise care in sepsis and enable, hopefully, most importantly for patients, swifter diagnosis and a faster recovery. Going forward, I'd like to take you through some of our published data and our future studies which we're trying to address specific questions. We were incredibly lucky to host a key opinion leader event in Athens in September last year, and as a doctor who's always been interested in sepsis, it was amazing, a little bit intimidating as we had 7 professors of intensive care and sepsis in the room, and there was a really good and robust and detailed discussion about ways forward, and everyone could see the potential for the H3.1 assay, and that very much those discussions have informed the design of studies going forward. We've got 2 papers published thus far and a lot more in the works. The first paper was the Moremont paper published in July 2022, and really that showed that H3.1 correlated with the severity of sepsis. In particular, it correlated very well with the H3.1 levels, correlated well with the SOFA score. And if you remember, I mentioned the SOFA score a little while ago as really a research tool for just assessing how sick people are with sepsis. The next paper was published by Professor Gael Forterre's group at just before the European Society of Intensive Care Medicine meeting in October. And that showed an association between H3.1 levels and mortality in patients with septic shock. Those patients who were sickest, those patients who were more likely to die, had the highest nucleosome levels. And actually we could see a change in trajectory of those patients who were going to survive. Those patients who survived, their net levels, their H3.1 levels, fell more rapidly than those patients who had a worse outcome. You heard me mention a few moments ago our novel, rapid, high throughput method for netosis induction. We're really excited by this paper and actually tremendously proud of our scientific team in California and the work they've done to identify triggers to stimulate neutrophils in a reliable, reproducible and consistent fashion, and actually to show the release of neutrophil extracellular traps, and then show how H3.1 correlates extremely well with the release of neutrophil extracellular traps. This is actually a key scientific plank for Volition. It underpins the validity of the test and shows that H3.1 does correspond with neutrophil extracellular traps. We've got 2 more papers we hope to publish to reinforce and reiterate the scientific validity and actually the unwinding of DNA in the next 12 months. What are we doing to prove the clinical utility? Well, we have 2 large studies ongoing at the moment. The first is with the Mars study group, and the second is with the German SEPNEP study group in Jena. Together, this will look at the evolution of sepsis in around 2,500 patients. So we're really excited to put H3.1 to this test. Again, we hope to publish this data in the autumn or perhaps early in Q4 of 2024. Finally, we have a study open at Guy's and St Thomas's Hospital dealing with fresh samples We aim to recruit around 500 patients, 50 patients undergoing cardiac surgery, and then 450 patients with sepsis, and to compare the two and see their evolution. Again, the questions we're asking there: how good is H3.1 at discriminating illness? How good is it at identifying severity of sepsis? And how good is it at tracking, following the trajectory or the severity of a patient's illness? We expect to finish that study in Q2 of 2025. So we've got a huge amount of work underway to really reinforce and prove the clinical utility of the test, combined with our refinement of the manufacturing process I mentioned earlier. We hope to have much of this data available for a data room at the European Society of Intensive Care Medicine Congress in October 2024. We're going to look at key measures: the disease severity, ICU mortality, ITU length of stay, 28-day mortality, duration, severity, and total volume of organ support required, and length of stay. As you heard me mention at the beginning of the talk, 80% of patients develop sepsis outside of hospitals. So our work is looking at patients presenting to the emergency room and to intensive care. It's not quite so focused on geography because we're taking our care to the patients. We're hoping to show its predictive value, its prognostic value, and potentially its therapeutic value too. These studies are across a variety of clinical settings, across a variety of patients. The aspiration here is to show its depth and breadth of use in clinical scenarios. And hopefully, if we can produce good data, that will speed and enable rapid adoption and rollout to clinicians across the world. Thank you for listening, and I hope I've shared some of my passion for sepsis and trying to improve outcomes for these patients, or for our patients. I'd like to hand you back to Gael now to take you through the commercial strategy. I'll be here till the end, and I'd be delighted to answer any questions anyone might have.
So thank you very much, Andy, for your presentation. So now a few words about our commercial strategy. As you know, we are focusing on the R&D at core. This is something we're very strong. We're doing it internally as well as with selected partners. And then when the product is ready, we are working with global players for the distribution side. In certain markets, whether it's fragmented or it makes sense, we might also add some regional companies. So we partner with those established diagnostic companies to sell, to process or test. And the goal here is really to leverage their network and expertise. And as well as, as the two principles that we outline, make sure we're not scaling our OpEx massively. So effectively, the model that we're working with is low CapEx for partners. And I'll mention in the next slide, again, why that is the case. And low OpEx for us because we're relying on the distribution partner for the scaling of our products. And then the second key principle that is very important to us is to be accessible price-wise, but also accessible worldwide. So now the building blocks of our net strategy, if you will. How do we build up the value for commercial partners and for the clinicians? So first, the platform and the product. We're effectively working on the transfer of our technology to different platforms. So that's linked to what I was mentioning about the low CapEx. Our partners don't have to onboard new machines. We're working with their existing system that also help accelerate the scalability of our product. Then we're working on the improvement of our product. As always, we incorporate the feedback of the clinicians, of the partners, and we iterate to bring to the market what we think is the best product. Market access. So this is the continued nurture of KOLs, networks, and then the centers of excellence that we mentioned. So we held a key event last year. We have another one coming in September 2024. This is really building the connection with the key people in the sepsis world, and it's yielding a lot of benefits to Volition. So the third building block, if you will, is regulatory. So now we have a clear pathway for the US 510K. In the EU, we have already an IVDD, and we will look into the timing of applying for IVDR. And then finally, and linked to what Andy was discussing, we're really building the clinical evidence of our product. And this will really translate in adoption by clinician, as well as build up the value for the commercial partners that we're discussing with. So the commercial strategy, based on those building blocks, and as I mentioned earlier, we're really focusing on licensing. That doesn't mean we don't do a couple direct sales with the centers of excellence, but really at core, we're focusing on building, a strong distribution partnership. You heard about the Heska No Mars agreement. It had the component of an upfront of milestone payment. So, when we sign, we receive 10 million upfront with Heska, again, no Mars. We received recently the 13 million milestone payment, and we have more coming. We also are working on a different structure around royalties as well as the sale of key components. So you will see in most of the transactions we do, at least 3 of the 4 components that I mentioned, the upfront, the milestone, the royalty, as well as the sale of the key components. But effectively, once we license a product, we are, receiving those payments on at the beginning and during the regulatory journey, if you will. But we also have ongoing sales, and I think that's really important. We're not giving away the product. We have this tail of sales and growing sales. Now, in terms of strategic partnership, we initiated a partnership with IDS. So this is a technology partner. Now it's moving into a distribution agreement. IDS has an install base worldwide, but here we're focusing on the EU. It's a outreach program, early access. We can call it a pilot for now. And subject to the feedback and the progress that we're making, we are hopefully going to dial up this activity. So I can make a little comparison. For those familiar with our vet product, we had this kind of similar initial partnership with Texas A&M. We're applying some of the same principles here. Now, in terms of key milestones that you can expect over the next year, as I mentioned, this early access program with IDS, we also have the centers of excellence. Some of them are going to convert now into routine. We are going to, based on the feedback and as well the timing of some negotiation, roll the same program in the US, but this time not as a regulated product, but as REO. This might take a little bit of time, so it could be later in the year, as well as in Asia. We're working on a new generation to incorporate the feedback that we got from partners of the product. So that's what we call the Gen 2, a lot of incremental improvement that help with the workflow of clinicians. It's not as much the performance, it's really focusing on the workflow and making it easier for clinicians and for commercial partners to onboard. Then we have the core of the discussion today, the clinical utility and demonstrate with strong data. So June or mid-year is kind of a turning point for us. We will hopefully have established the cutoff, the value proposition, and it will be really the time for us to push with commercial partners. We're discussing with them already. Some are very interested, but we need, additional data to really crystallize the value. And just to clarify, the Gen 2 is what will be used for licensing and the pivotal and regulatory studies for the one we might be conducting. Obviously, for the partners, it will be their version of our products. And then finally, I think in October, you can expect public presentation of this data at the ESICM. Looking forward, we're excited to launch our Nu.Q Net product. It's a low-cost, rapid, easy-to-use, and accessible test to help save and improve millions of lives worldwide. So with that said now, I'll turn over back to Sue for question and answers. Thank you.